It's my pleasure to introduce our first speaker this morning, Dr. Lloyd Fricker. And anybody who has serendipity in their title, I'd like to know I'm looking forward to what he has to say, so please help me welcome Lloyd.
By thanking Larry for the invitation to come here. I've really enjoyed the presentation so far, and I hope I can live up to the high standard that's from yesterday's talks. I met Larry at a meeting in Australia on peptides. It was a wonderful meeting, but not nearly as stimulating as this one. So, but it was a nice location on Hamilton Island. Okay, so I thought I would, well, first of all, the bio blurb that I sent in apparently was too short, so apparently Carissa overlooked it. I'm not the CEO of any companies. I'm a professor at Albert Einstein, and I didn't proofread what she sent me. It's my fault. So it doesn't say what I intended to put in the bio blurb, which is that the facts. I have a PhD from Hopkins from 1983. I stopped by Boulder. That wasn't in the bio blurb, but I stopped by Boulder on my way out west to do a postdoc, and then I've been at Albert Einstein for 25 plus years. I wear three hats there, I guess, or I do have three jobs. One is a scientist and professor. The other is I am a pharmacology course leader, so I teach the medical students all about pharmacology. And the third thing is I'm a writer. Well, all scientists are writers. We write papers, but I do creative writing, which shouldn't be scientific writing. And we've been talking about success, that everybody has solutions. Success is really easy. We just go up a few steps. How do we succeed in improving healthcare? It's just a few steps up. This is actually one cover from one of my books. Every writer I've seen usually puts their cover on their slides. So, succeed in science and avoid getting a real job. It's humor and serious advice mixed together, and I don't differentiate the two in places, so you're on your own trying to figure out what they are. I have another earlier humor book on writing bad grants, and other helpful advice for scientists. And then a third book, which is somewhat creative, but it's more serious. It's on peptides, and that's about to be released in a couple weeks. And a few more book projects. So what I'm going to talk about is first, what are peptides? How important they are in general, what are peptides, and how they are signaling between cells as neuropeptides and peptide hormones. And then some new ideas for what peptides can be doing inside a cell. That is the twilight zone, and if it's real, it's going to be paradigm shifting, I believe. But I'm not sure it's real yet, so I want to put that disclaimer out there. It's a great idea, and might be true. So then I'm going to talk about drug discovery a bit, and how peptides are integral in drug discovery, and just the serendipity of drug discovery, rational design, or as I like to call it, intelligent design, versus random chance and selection, which is more like evolution. Most drugs, if not all drugs, have some amount of random chance in their discovery, some amount of selection. Even if you think you know the target, there are plenty of examples of things that didn't work as planned, or things that worked differently than planned. Some classic examples: isoniazid was developed as a TB drug, anti-tuberculosis. And what do you do when you have a drug? Is you derivatize it. So the chemists made iproniazid, tested it out in TB, and it worked about as well as isoniazid. But the patients on iproniazid were really happy. Now, as I heard it when I was told this in pharmacology many years ago, I was told that you would be happy too if you had TB at the time and it was being cured, because that was an incurable disease. It was a death sentence, and if it's being cured, you'd be happy. And the people on isoniazid were happy, but the people on iproniazid were happier. And so chemists went in, or biochemists went in, looked at what it was doing. It inhibited MAO, monoamine oxidase. And that was the start of the whole antidepressant monoamine oxidase category of drugs, some of which are still used today. Not a frontline, but certainly an important class of drugs for depression. Chlorpromazine was an antihistamine. It is an antihistamine. It was developed as an antihistamine, and as you know, many antihistamines like Benadryl are sedating. So they had some manic schizophrenic patients and decided to sedate them with chlorpromazine. And they left them on the chlorpromazine for several weeks. And after several weeks, they got better. The voices in the head stopped. That was weird. So they went back and looked and found out it was a D2 dopamine 2 receptor antagonist. Led to a whole new paradigm for antipsychotics. That was ancient days. Of course, nowadays we know how to design drugs like Viagra, which had a bit of a side effect in men. And it was being developed as a heart drug. And minoxidil was being developed for something else, and it caused hair growth. That sold as Rogaine. So serendipity is with us, and it's part of the integral process of drug discovery, even if we like to pretend we know how to design drugs and avoid this random chance. I'm going to talk a little bit as a side theme about just hypothesis-driven research and serendipity. The classic example of non-scientists think that we do, scientists do, is that we start off with a hypothesis. We start off with an observation, we make a hypothesis. From that hypothesis, we then predict things, test it out, and either confirm, find what we were looking for, or we reject our hypothesis. All scientists do that some of the time, and some scientists do this all of the time, but all scientists don't do this all of the time. We would never really get anywhere if we did. The true paradigm-shifting discoveries come when we're not testing our hypothesis, or we test our hypothesis and find something we weren't looking for, something completely different, something we couldn't have hypothesized because we didn't know it existed. So that's going to be a side theme. Serendipity in drug discovery, serendipity in basic research. And a big buzzword is translational research. Everybody is for it, and I'm certainly for it too. But I'm also proposing we think about a term, it's been around forever, but nobody uses this term, retro-translational research. That's what they did with the chlorpromazine and the iproniazid. You find something that doesn't make sense, and you go backwards into the basic science. It's not true, unbiased basic research. You're not just trying to find fundamental mechanisms. It's targeted for that disease and that interesting process. So it is translational, but sometimes you have to go backwards. You couldn't sell a car in America or anywhere if it didn't have reverse. Sometimes you have to go backwards before you can go forwards. So that's going to be another sub-theme, and I think that is an important thing. You can't talk about healthcare without talking about drugs. Drugs are an integral part of healthcare that make up about 10 to 15% of the expenditures, and it's certainly an important part. And if they're used correctly, they can save money. They can keep people out of the hospital, which is very expensive. If they're used incorrectly, they can cause people to go to the hospital. 5% of hospital admissions are ascribed to adverse drug reactions. So understanding drugs is, of course, important. So I'm going to talk about peptides and drugs and mix it all up. What are peptides? Well, they're chains of amino acids, and amino acids make up proteins as well as peptides. In the laboratory, you can make peptides. Chemists can make peptides from the monomers, from the amino acids. But in most cases, biologically, peptides aren't made this way. There's a few examples. Glutathione is one, where peptides are made by amino acids being stuck together. But usually, in the biology, the peptides are made from proteins by digestion, by proteases. And then to complete the cycle, and it's a very efficient cycle, the peptides are digested back into amino acids to complete the recycling, and then they're made into proteins again. So I'll be talking a little bit about the peptidases involved. So what are peptides? Biological peptides, there's really three categories. Those involved in cell-cell signaling, such as peptide hormones. You've all heard of insulin, of course. Neuropeptides, like endorphin, the endogenous morphine in the brain. There are other types of cell-cell signaling peptides, chemoattractants and growth factors. Anti-defense or defense mechanism peptides. We have antimicrobials in our body that keep the gut microbes in check. We need them in our gut, but we don't want them to get too close, so we secrete things like defensins and other molecules into the gut to keep the bacteria from overgrowing. There's toxins not in humans known, but in like snakes and lizards that are important, and some pharmaceuticals have been based on these important toxins. And antigen presentation is also a defense mechanism, and it involves peptides. There are some cases of intracellular signaling of peptides being cleaved off of proteins and going to the nucleus and turning on genes or turning off genes. In addition to these endogenous roles, a lot of people use synthetic peptides to modulate protein-protein interactions. Proteins have to bind to something else to function, either as RNA or DNA or quite often other proteins. Most proteins work as complexes of multi-protein complexes. They have cofactors or binding proteins. And biochemists have peptides synthesized to mimic the binding site of a protein-protein interaction and add the peptides to the cell, somehow introduce it, get it in. And this interaction can either mimic the protein-protein interaction or it can block the protein-protein interaction. And by doing this, one has very specific tools to affect that protein-protein function. Chemists or biochemists use this frequently. There's hundreds, probably thousands of papers that use this. But nobody thinks that this is endogenous. People think that scientists invented this great idea, But it's hard for me to imagine that Mother Nature hasn't already figured this important process out, and that this, in fact, could be endogenous. And this is where I'm going to get to in the end of the last half of the talk. So first, let me focus on peptide hormones and neuropeptides. They're involved in a lot of processes. If there's something that isn't on the slide, it's probably because I forgot to include it. One could just say they pretty much do everything. And many of these are good targets for drugs. Of course, feeding body weight, that's a multi-billion-dollar a year industry. Other things, mood, anxiety, stress, these are all ideal targets for drugs. Now, I was reading a review by Flora Strand, wrote a book on peptides, and the introduction to it, and this nice quote. I'm not sure what the three deadly sins are. Certainly, gluttony would be one, because of feeding body weight. Sloth, I guess, would be sleep-wake cycles, and maybe envy, and there's probably more than 3 of the 7 deadly sins, but I don't remember what all of the 7 ones are. So they do everything. They're important, and they differ a little bit from neurotransmitters. Neurotransmitters are doing the heavy lifting of the signaling between cells. Neurotransmitters like serotonin, dopamine, acetylcholine are secreted from a cell. They cross the synapse, stimulate a receptor, and then they're recycled. They're taken back up either whole, like serotonin and dopamine, or they're broken up, and then the pieces are taken back up, like acetylcholine. And this allows them very efficient synthesis, very efficient termination of the signal by reuptake, and that's the major cell-cell signaling molecules are these neurotransmitters. Peptides take longer to be made. They have to be made from proteins in the endoplasmic reticulum. Well, first DNA to RNA, then RNA to the protein, then the protein is chopped up into peptides, and those are secreted and broken down, so you have to start again. It's like a wine, a fine wine. You don't start and make wine when you need it. You make it, well, years in advance. In this case, it's hours or days in advance. So it's not really the fast-acting neurotransmitter. That's these guys. These are more the slower things, like feeding isn't something you have for a nanosecond or a millisecond. Feeding is a thing you feel for minutes or hours. So these kinds of peptides are more involved in tuning the fast-acting neurotransmitters that are going through here, this pathway. And it's much more complex than one peptide, one receptor. There are probably no examples that I can think of that are one peptide, one receptor. In most cases, a peptide binds to multiple receptors, usually with different affinities and producing different effects. Or multiple peptides can bind to one receptor, or both. Multiple peptides bind to multiple receptors, and that's really the norm. It's a little bit more complex than the one peptide, one neurotransmitter, one receptor. Peptides are made from proteins, and the proteins give rise to multiple peptides from that same protein. In many cases, all of the products, or half the products, are bioactive. The other half might not have activities known, but the protein precursor rarely just makes a single bioactive moiety. Usually it's multiple things. Now the enzymes that make these, this carboxypeptidase E is an enzyme I discovered 30 years ago as a graduate student, and it makes pretty much every neuropeptide in your body, every peptide hormone, things like insulin. It doesn't make the C-terminal piece, though. It makes all the other pieces. And then there's an endopeptidase, actually 2 of them, that process. I'm not going to talk more about these enzymes. But let me try to reduce it down to something that I think makes it more easier to understand, is that peptides are like words, or a short protein is a precursor of a neuropeptide. It's typically 200 to 300 amino acids. A tweet is, I don't know what, 140 characters or something. So it's roughly similar. Words are made of 26 letters. Peptides are made of 20 amino acids. Of course, there's uppercase and lowercase, and there's little, for foreign languages, there's little things that go on the letters. So there's more than just the 26, but same with amino acids. There can be phosphate stuck on, sulfate stuck on, carbohydrate. So you have ways to vary it, just like those tildes and things. Small changes in words can alter meaning. Stop versus step, of course. Small changes in the peptides can alter meaning. Order matters. Of the letters, stop versus spot means completely different things. Same with peptides. If you scramble a peptide, it doesn't mean the same thing. Single words have meaning, stop, versus combinations of words, don't stop. It means something completely different. Likewise, peptides also produce their effects in combinations that can change. And this is a factor in thinking of peptides as drugs. Is often people focus on the one peptide from these precursors, not thinking that the peptides are made as a team and secreted as a team and function as a team. And the drugs are often tested out based on that one peptide working as if it's the only show in town, but it isn't. And processing can alter meaning. Nowhere versus now here. It's the same thing. You just break it up, you get two different meanings. And processing of peptides can also matter. This is a peptide found by a friend of mine in Brazil, Imer Ferro. He found this peptide. It's a peptide that works at the THC receptor. I know some of you are thinking, wait a minute, those are lipid molecules. Well, peptides can also bind to that receptor. These are endopeptide cannabinoids. And this blocks and this stimulates. A small difference, whether it's processed here or not, affects whether it's an agonist or an antagonist. Okay, so I was interested in peptides in the late 70s and went to Hopkins to work on them. And this came out in 1980. It reflected the buzz at the time. The world's most amazing drugs that are going to be better than aspirin, you beat pain, insomnia, overeating. It mentions many, many things that it does. And the little small print there is that these things are based on peptides. So there's been a lot of hype. Where are the drugs based on peptides? There's only a handful that have been developed. There's certainly not as many as people had expected. And I'm going to allude to why not. But let's just talk about all drug discovery. This is, you saw this, a little bit of this data. Garrett showed just from 97 on. He didn't show up to 96. From 1970 to 96, the number of new drugs, all new drugs approved, not based on peptides, but all new drugs, was increasing. And if you continue this line up, it's incredible. You're going to have 50 to 100 new drugs a year by 2012. Then if you include the data from 97 to 2007, you see an opposite kind of curve. And you would extrapolate this and say, well, we're not going to have 50 to 100 drugs. We're going to have 0 to 10 new drugs a year. What's happening? The money's going up. The number of new drug discovery isn't changing, is decreasing. If you add the newest data, and I had to change scales here because the FDA website didn't have the same data from the Goodman and Gilman, and I think they counted differently as what is really truly a new drug. Is it a new formulation or a true new molecule? It's really held flat despite the money increasing. And it could be that you just can't hurry luck. That there's serendipity involved, and no matter how much money you throw at it, you still need that random events and random chance and people to have their eyes open and notice things like, gee, these schizophrenics on this drug for 3 weeks aren't just sedated, they're not hearing voices in their head anymore. Is that included in clinical trials now? I don't know. Maybe not. So serendipity in drug discovery, let me just give you one example of a drug based on a peptide that was developed by Merck. It was based on substance P, which is a neuropeptide in the brain and spinal cord. It was thought to be very important in pain in animal studies. So a drug was developed that blocked it, an antagonist, MK869. In clinical trials, it worked about as well as aspirin. And nobody's going to pay $50 a pill for something that works the same as aspirin. So then they got the idea of testing it for depression, antidepressant. The receptors were in places that were involved in mood and thought to be important for depression, and it was tested. Animal studies looked great, phase 2 studies looked great, but phase 3 studies, not so good. It was no better than the placebo. So they kept shopping around, and they found it worked in emesis, vomiting, and it was tested, it worked well, and it's now approved by the FDA. So that's the idea of serendipity, just like the Viagra story. You start off with one thing, it didn't work, but you find it works for something else. Serendipity is an important part of basic science, too. Now, after I discovered CPE, the enzyme that makes all neuropeptides, I was working on it for many years. People were telling me I should make a knockout mouse so I could study it. And I told them, no, it's not going to work. It's going to be embryonic lethal, because if you don't have this enzyme, take out carboxypeptidase E, you're not going to have all of these peptides. You'll just have this one, but none of these. That's going to be embryonic lethal. Well, I don't want to study dead pups, mouse pups. I was right, but for the wrong reason. There was no point in making the knockout because it already existed. There was a spontaneous mutation in the early 1970s at Jackson Lab, and they noticed this mouse was fat and sterile. So it made it a little difficult to study. They had to do ovarian transplants to keep the line alive, but they did. And after 20 some odd years, the geneticist on the project, Ed Leiter, was mapping it to the carboxypeptidase E gene. At that time, he had maybe one of 20 genes, wasn't sure if this was it. called me up. Was I interested? Of course I was. I got some tissue. The next day, found out there was no activity, or very little activity, CPE in these mice. So we went ahead, they found a mutation, we made the mutation, it made the enzyme inactive, it was great. It was like a knockout mouse. And everybody wanted to know, well, the processing intermediates accumulate, the mature forms decreased, we looked at that. Everyone wanted to know, why are the mice fat? Great question, I'm gonna get to it in a few minutes. I wanted to know, why are they alive? They should be dead. You know, you don't have this critical enzyme. Either neuropeptides don't really matter, which it wouldn't be good for those hundreds of us that are working on neuropeptides, or perhaps there was something else going on. Is there another enzyme in the pathway? So I went looking, and within a few months we had found another enzyme. We did see that there was very low levels of these mature forms you could detect at low levels for many of them, suggesting maybe there was another enzyme in the pathway. We went looking, and we found first this one enzyme we called carboxypeptidase D. It was also found simultaneously by a group in San Francisco looking for the hepatitis B virus, for duck hepatitis B. It was found in a Drosophila lab looking for the silver mutation that caused Drosophila to have problems. So it was found 3 different systems all at the same year, and we called it carboxypeptidase D. We also found 11 other carboxypeptidases that I'm not going to talk about. Some of them look like they're interesting targets. One of them, very recently, I got a phone call, actually an email, from a geneticist that mapped a gene causing human epilepsy to one of these other carboxypeptidases we had found, CPA6. Now we're trying to figure out, retrotranslational again, how this mutation could affect the body to give rise to a predisposition to epilepsy. So I won't say more about these enzymes because I'm going to focus on peptides. So the question is, why are they fat? So to look at this, we developed a new technique based on the proteomics techniques, where you take samples from the different animals, you label them with isotopic tags. These are chemicals that look identical chemically, except they have different amounts of deuterium or C13 or N15. You combine the samples and pool them and then analyze by mass spec. And the mass spec is so sensitive that difference of several deuteriums versus hydrogen makes the mass a little bit heavier. And so you see, instead of one peak for the peptide, you see peak pairs. And they're either the same height or one's higher than the other. It tells you how much relative peptide is in the 2 samples. This is wonderful. You don't have to look for just those small number of peptides that you know about. You can look for new peptides as well. You can look for hundreds of peptides in one experiment. Let me just show you a little bit of data rather than everything be summaries. We actually use this TMAB label. Chemist Fred Rainier in Purdue designed the label for protein analysis. He designed the D0 and D9, and we filled out the series by making all 5 of these with different deuterium numbers on this group here, or C13, or both. And here's actual data. You can do a run, you see a bunch of peaks, you blow this up on the computer, you begin to see that each one is separated into 5 peaks, and then you blow it up further, and you can measure the peak height of all 5 of these, and you know how much peptide was in each of those 5 samples. And this is just 1 second elute from a 20-minute gradient. If you look just a little bit earlier, instead of 20 minutes now, 19.9 minutes, you see a different set of peptides in the same mass range. If you look just a little bit later, you see a whole different set of peptides. So over the 20-minute gradient, you see hundreds and hundreds of peptides coming out, and you can quantify each one of them. This isn't 10 times better than a radioimmunoassay. This is 1,000 times faster than raising antibodies to each peptide and measuring them by radioimmunoassay and characterizing each antibody. It's also better in that you can look for things you don't know existed. So this is the list. Everybody that does proteomics or peptidomics is obligated by law to show a list of all the things they found. This is my list. You're not expected to read it. But the top of this list is this thing called ProSAS. Nobody had known of ProSAS before. We would never have found it if we were focusing on what was known using radioimmunoassays. But we found it, found 33 different peptides from it, and it represented 17% of all the peptides we saw. It's a major, what's the most abundant, or one of the most abundant neuropeptide precursors in brain, and nobody knew about it. So what does it look like? Here it is. Here's the peptides we have. It doesn't matter what the names are. The names are based on amino acid sequence. And there are some cases, as I was saying, where words are processed differently. You get different meanings. Well, here, you can either have a big form or a little form, depending on if it's cleaved here, same out here. You get big or little SAS. So the key thing is, what does it do? Well, how do you know what something does? We heard about yesterday morning antibodies and distribution is a key thing for what something does. The other two ways that basic scientists figure out what something does is what happens if you put more of it into a biological system, and the other thing is what happens if you take it out from the biological system. So what happens, and let me just summarize, the distribution is neuroendocrine. It's not in liver or kidney, it's really neuroendocrine specific, and in fact it's highest in the hypothalamus and parts of the brain that control body weight. What happens if you put too much in, overexpress it? Well, one of the mice, was done together with Amgen, one of the mice got up to 70 grams. She was expressing 5 times more. That's like a sumo wrestler, you know, that's like me being 500 pounds. But normal mice expressing more modest levels, 2 times the baseline, were only about 30 grams, so about 20% overweight. And if you knock it out, the knockout mice are underweight. But they are also anxious. So I'm not going to jump up and down and say, this is the next drug for treating body weight because you'll be skinny but anxious, and that's not what's going to be useful. But when you knock something out, you're knocking out all the pieces, the entire sentence, the entire tweet. But if you just take out parts of it, an agonist or antagonist, an antagonist that blocks the receptor could just take out selective words. And we tested antibodies to these, injecting them into brain, and found out antibodies to big Len and pen blocked feeding, but not little n or SAS. So there is hope that if there are specific receptors for these forms, we could not knock the whole thing out, but selectively with the drugs block some of them. So there still is hope. I'm not saying it's doomed, but the anxiety is a big problem, so we think it's involved in body weight, anxiety, and many other things, actually, sleep-wake cycles as well. So why are the mice fat? Let me briefly go through this. It's a bit of data, but the bottom line is you're fat if you eat more or burn less, metabolize less. Eat meaning eat and absorb into the body. If you don't absorb it in the intestine or stomach, it doesn't count. So if you absorb more and you burn less, you're going to gain weight. If you eat less and burn more, you're going to lose weight. There are the big players. Every review on peptides and body weight talks about neuropeptide Y and alpha MSH as being the satiety peptide and orexigenic peptide. So here's a bit of data. Ideally, every mouse, if you compare a wild-type mouse in using this peptidomics relative scheme, would be exactly 1.0, but they're not. Even though they're genetically similar, there's still mouse-to-mouse variation, and so we see a bell-shaped curve for wild-type versus wild-type. But when we look at fat versus wild-type, we don't see a bell-shaped curve. We see an inverted U. Either things are way up, greater than 2 ratio, or way down, almost undetectable, and very few things are in the middle. So what are the things that go up? Well, things that go up are these peptides with the large ARG and lyserge. All of these that go way up have a C-terminal basic on them. They're the intermediates. Things that don't change are the C-termini. That's what you'd expect, because CPE is not needed for that. Things that go down are the things that do need CPE. Either it's only CPE or it's a combination of CPE and D that make it. So why are the mice fat then? Well, alpha MSH and NPY are both in the group that go way down. So you're losing alpha MSH and NPY. But big Len, that's the C-terminus, remember? And so that doesn't change, actually. That goes up a little bit. It might be upregulated, in fact. In the hypothalamus, it went up a bit. So why are they fat? I'm proposing that it's because they lose the brakes, and they lose some of the accelerator, but not all of the accelerators. It's stuck on, and it's like an old car without brakes, but it still works, and it's going to crash. So they get fatter. So from Wikipedia, the phrase, "may you find what you're looking for," is attributed to an ancient curse. And this brings up a couple issues. Why is it a curse? I'm in a session that's on education, so I thought I'd just give a little rant about education. I think we're all born as scientists. Kids start off as scientists. We're naturally curious. And those of us that go into science are those of us that haven't had the curiosity beaten out of us. Science classes in high school and grade school are pretty brutal. It's memorizing things, memorizing parts of your body or names of parts of a flower or the species. It's not what scientists do. Scientists discover things. Kids start off discovering things. First, they discover conservation of objects. You can play peekaboo, and it's fun because mom still exists or dad still exists when your eyes are closed, and they're still there. Conservation of objects is something that we discover as scientists when we're young. We discover gravity. We take Cheerios and drop them off the tray on the side. They go. And we take the whole bowl of Cheerios and drop it off with the milk and the spoon, and it goes, and then Dad gets really mad, and you don't get more Cheerios for a while. So we discover things as scientists, but it gets beaten out of us unless, most of us that are scientists, unless we've had somebody show us that science isn't just memorizing things. It's fun, it's doing, it's exploration. We don't expect kids that want to draw. Before you give them a box of crayons, they must memorize 256 Crayola colors and memorize all these facts about all the famous painters that have come before and every famous painting they've made. And finally, after they've memorized these things and they're 22 years old and in grad school, you say, now you can have the crayons. We don't do this for kids that want to play sports. You can't get a ball until you've memorized all the rules of all the games and all the famous sports players and their stats for the last 50 years. And then we'll give you a ball. But that's what we do for science. We make it as if you have to study all this stuff, and then finally in grad school, if you haven't already discovered it, you discover research is a lot of fun. Most of us, though, have discovered it earlier. So since I was put in this session on education, I thought I'd throw that out. Let me also go into another thing about finding the obvious. So with peptidomics, you find things that you're not looking for, things you don't even know existed. So let me give another thing. A famous discoverer, wasn't a scientist, was Columbus. Christopher Columbus. What did he discover? He discovered a new route to India. That's what he thought. He came over, he found some plants. He said, these plants seem like tropical plants. They smell like cinnamon. It must be in India. He found people with brown skin and straight hair. This must be India. He didn't recognize that this was a Stone Age society he found, and India was a very sophisticated Middle Age society, not at all close to the grass huts. But he was convinced. The maps he had didn't look anything like the maps he had of the islands off India, but he was convinced that he had found India. And he made multiple voyages, and he went each time back saying, I found India. I found a new route to India. So we tend to find what we're looking for as scientists, and we tend to ignore the things that don't fit with what we're looking for. And we did also, like most scientists, ignore the things we didn't think fit our model. About half the things we saw were in the secretory pathway, and that's what we were looking for. Neuropeptides, or new neuropeptides, which we had predicted, but we didn't predict that we would find peptides inside the cell from cytosolic proteins, because the dogma was that they don't exist. The dogma is that if you put a peptide in a cell, it will be degraded within 7 seconds. There were some studies that showed that with a handful of peptides. But we were finding peptides from inside the cell. Could they be functional? These are peptides from cytosolic, mitochondria, and other proteins. Could they be functional? So a friend of mine in Brazil, Emer Ferro, had been proposing for about 10 years now that they might be functional. And after many beers and discussions, we put together this model where the proteins are broken down by enzymes like proteasome and galpanes. And then the peptides do exist. They're not just rapidly taken out by the aminopeptidases and endopeptidases. Some do exist. And there are many processing enzymes in a cell that are not degrading enzymes. Their function has been a mystery for decades. They take a peptide and convert some of the peptides into smaller forms. Why? Why would the body go through this trouble to make all these things if they're not doing something functionally? And then it's well known, as I said earlier, that peptides can be used, synthetic peptides can be introduced into a cell to change protein-protein interactions. And it's just inconceivable to me that Mother Nature hadn't figured this out and uses some of the peptides. So this is the hypothesis. It hasn't been proven yet. There's some evidence to support this, but it's really the twilight zone. I don't want you to come away from this thinking, oh, this is a new thing. It's a new model. And that the endogenous peptides affect intracellular processes, which is like microRNA, which for many years was thought to be junk, but now is thought to be a very important way that small pieces of RNA can regulate the big pieces of RNA. And the same thing, small pieces of proteins, small peptides, can interact with the larger proteins and do functions. Let me just briefly end with a little bit about the proteasome. Now, the proteasome is an enzyme that is a complex. It has 3 active site enzymes that cleave proteins and many other subunits. And it chops up the proteins into peptides. Now, there's a drug, Velcade, bortezomib. It's a generic name. It's a proteasome inhibitor. It's most active at the beta-5 sites, which is the chymotryptic-like, and it also cleaves the beta-1 subunit, which has been said to be caspase-like. But the mechanism of action isn't well understood. Even though it clearly is a proteasome inhibitor, how does that really change things in the cell? And this is a reference from a paper of Goldberg's that the quote is, an important question is how low concentrations of the inhibitors can block cell growth and cause apoptosis of myeloma cells at concentrations where proteolysis is reduced by only 10 to 25%. That's a modest change in protein turnover, but it produces a big change in the cell. It kills the cell. So we thought, what would happen if we treated cells with bortezomib? What would happen to the peptidome? And this is a heat map where the red, bright red, is things that go up 5-fold. Bright green are things that go down 5-fold. And gray is things that don't change. This is the dose. You're not expected to read all of this, but the low dose for 1 hour, 5 nanomolar, not too much change. You go up 6 hours. By 16 hours, you're starting to see some changes. But with a slightly higher dose, 50 or 500, now these aren't out of the physiological range. That when people take it, it's hard to calculate what the cell is actually seeing, but it's somewhere in the 50 nanomolar level is a realistic estimate, 50 to 100. So this is somewhat physiological, 50 here. And you see a lot of changes. In fact, almost everything changes. It either goes down or it goes up. You're blocking the production of the peptides by inhibiting the enzyme subunit that makes it. But now the one that isn't being inhibited can process peptides that it didn't normally make, so that's why some things go up. And so the hypothesis is then that it could be the change in the peptidome, which is changing mostly 5-fold, 500%, opposed to a change in the peptide or protein turnover that's only changing 10 or 20%. It could be this change in the peptidome that's the real mechanism of action. Well, that's not true. I mean, the mechanism of action is proteasome inhibitor, but how does that translate to the use as an anti-cancer drug? This could be the next step, and that would be important to know that. Obviously, you need to know how drugs really work before you can translate new developments in them. So that's the retrotranslational. And, I did protein network analysis, which I'm not a big fan of these interactomes. But of these 49 proteins that we found in the cells that changed with bortezomib, 47 of them were in a network using this Ingenuity systems pathway analysis. And the network was involved in cell growth, proliferation, and death, which is exactly what should be changing if you're inducing death in these cells. So just to conclude, I've talked about the classic neuropeptides that are secreted, function at the next cell. I didn't talk about this class, which would be these intracellular peptides that can be secreted. Some of them are secreted, and some of them stimulate G protein coupled receptors or have other targets. So there's also this middle pathway of this fusion between these two. But then I talked a little bit about this intracellular signaling pathway, which is, once again, not proven, but it's a great idea. And the idea that the proteasome inhibitors certainly alter this peptidome, and these changes might have biological effects, or maybe they're contributing to the side effects of some of the drugs. So just to leave it with the thought, so this has been the drug discovery rate of new drugs over the last 40 years. This has been the appearance of the word translational research in either publications or NIH grants. It's been an explosion of this. And I don't think this caused the drug to fall off, because it would have had to be out here, and it just really started. It's been really taking off. So what I'm sort of speculating, this is my last data slide, or summary slide. The discovery of most drugs has involved serendipity. That's certainly true. Basic science is important, especially retro. Let's get back to the mechanism and find either new drug targets or find out how drugs really work. And a question, I'm not really sure of the answer. We certainly need translational research. I'm not trying to say it should all be backwards, but will this emphasis on translational research actually lead to an increase in new drugs, or might it inhibit it if we're not also including retro-translational research? If we're taking away from retro-translational to focus entirely on translational, I would predict we would actually lose some of these basic things that will lead to the true new developments. And let me conclude. Everybody shows a picture of where they're from. This is Albert Einstein College of Medicine, one picture. But one picture is just one little slice. You have to take more pictures, and you begin to get a set of pictures. As you add more shots of Einstein, you get a different view of Einstein. Everybody asks, did I make this? Yes, I did. It took about 3 weeks of evenings, just a couple hours an evening. So it took a bit of time, but not any more than a jigsaw puzzle. So that's it. And I think I have time for a couple of questions. Thank you very much.